All right, great. And so next we heard a little bit about APOL1 and kind of some of these other influencing factors, but we want to make sure that everyone on the call has a background to what APOL1 is and what different touch points that can be used to address disparities that might be caused by APOL1 mediated kidney disease. And so I'm going to invite Dr. Hall to come and speak about APOL1, and I'll share a little bit about Dr. Hall's background. So Dr. Gentzon Hall, he's an assistant professor of medicine and nephrology at Duke University School of Medicine, and also the director of Blomkamp Foundation HBCU Outreach and Workforce Development. He received his MD and PhD degrees from the University of Maryland at Baltimore School of Medicine and completed his internal medicine residency and nephrology fellowship training at Duke University. He is faculty at the Duke Molecular Physiology Institute, where his lab focuses on the genetics of inherited kidney disease, with a special focus of protein washing kidney diseases that disproportionately affect African Americans. Dr. Hall's research has been funded by the National Institutes of Health, the Doris Duke Charitable Trust, the Robert Wood Johnson Foundation, the American Society of Nephrology, and Duke University School of Medicine Strong Start program. And his work has been published in top tier journals, such as the Journal of the American Society of Nephrology, Hypertension, the Journal of Clinical Investigation, and the Journal of Clinical Investigation Insight, and the American Journal of Physiology, Renal Physiology. He is committed to improving the health and well-being of medically underserved populations through translation of novel discoveries in renal genetics and the improvement of patient education and access to health information. So I'm going to call up Dr. Hall to share your screen and share a little bit more about APOL1-mediated kidney disease.
All right, good evening, everybody. On the East Coast, it is bedtime for my kids, so I'll just say in advance, there's someone who bursts into the room and gives an impromptu hello. It's just one of them. They're kind of in their bedtime routine. But, I think it's a fantastic way to celebrate APOL1 mediated kidney disease awareness day. I've been sort of following online different platforms, Instagram and so on. And last year was exciting to see there being attention brought to this issue of major consequence. And it's even more exciting to see so much more attention. I think, especially at the level of potential trainees and patients involved in managing uh, chronic kidney disease or advocating for others who are still dealing with it. It's a really important effort that I think is being rallied by the community, and I thank you guys for being a part of it this evening. Okay, so let me just see where we are as far as my presentation. I want to know what you can see. Are you seeing my presentation view or the presenter view? I may have to switch the display.
I'm
Breathe.
seeing, good.
Go ahead, Eunice. I'm sorry.
I'm seeing the slides in PowerPoint at the moment without
Okay.
notes.
All right. Let me see if I can get it to work now. Is that a change?
It looks the same to me here.
Looks the same. Huh. Okay. So it may just, like I said, I'm working from a few screens and it's, maybe I'm having a little difficulty here identifying the right one. 1 second. Give one more shot at this.
In the meantime, welcome to everyone that's just joining on. Feel free to place questions or comments in the chat as the presentations are going on. And I can see it now. This is perfect.
Perfect. All right. So talk today, it'll be APOL1 mediated kidney disease, current understanding and implications for health equity. Just at the outset, there's going to be a little bit of, sort of science here. If you're not comfortable, I've done the best I can to sort of focus your attention on the salient points. But for those of you in medical school, take this as an opportunity to see how far we've come in just a short period of time. I want you to understand that all of this development has happened in the time span of my training experience. So APOL1 was discovered in 2010 by an investigator, a premier renal geneticist out of Harvard, who reported it first to the world as an interesting outcome discovered in what we know as genome-wide association study. So you're essentially looking at a broad group of people with a significant phenotype and searching for a potentially, relevant genetic cause and or contributor. And the APOL1 was discovered in that process. Since then, 2010, I was a fellow. Now, as an assistant professor, I'm seeing the manifestation of that work in clinical trials. And so just, if you're kind of in any way thinking about the instability of the current environment, the challenge maybe to you professionally going forward, just know that science still moves on, that it is a continual pursuit of facts and evidence, and that evidence, once it's presented, vetted, tested, peer-reviewed, and released, it can't be challenged. And we use that type of information to base therapeutic decisions, develop new therapies, and it's still an encouraging place, I find, to be involved in patient care. So as you're hearing all of this, think about nephrology. That's my shameless pitch. All right, so here are my disclosures, and I just want to sort of give you a sense for what to expect. So, and you're seeing my objective slide, right? Okay, so we're gonna do a little bit of a review of essential concepts and terminology, not to bog you down, but just to kind of keep you in register as we go through the rest of the talk. We're going to describe the increasing prevalence of focal segmental glomerulosclerosis. That is an injury pattern, not as much a disease, but it's one of the hallmarks of AMKD that you will hear me describe in that way, FSGS, fairly regularly throughout the talk. And we'll talk about FSGS as a key driver of kidney failure in African Americans, and I'll provide you with at least a little bit of the evidence that's documented this over the course of about 20 years, the last 20 years or so. We're going to review the discovery of APOL1, renal risk variants, and how they sort of behave biologically. We'll discuss proposed mechanisms of how APOL1 renal risk variants actually injure cells of the kidney filter known as podocytes. We'll discuss them shortly. We'll discuss emerging therapies and clinical trial opportunities that really have, in the past few years, presented a lot of hope, at least on the potential for rational treatment of something that is certainly a hardwired genetic disease, if you will. And finally, we'll discuss EMBARK with a little bit more body and just sort of give a sense for what we hope to accomplish, how the program is structured, and where you can get involved. Okay. So essential concepts and terminology, we're going to start off to the left with this graphic. The nephron is such a beautiful structure. There's a lot to discuss about it, and in your lectures on renal physiology, you will have more than enough to consider from countercurrent multiplication and the filtration system. But we're going to focus on this small area of the nephron, the glomerulus, which is a network of capillaries, and they are surrounded by a very special cell called a podocyte. And that cell is responsible for a variety of things that range from early development and the ultimate shaping and architecture of this filtration unit, and as well as the ultrafiltration apparatus, a very fine, almost mesh-like region that allows the filtering of waste products out into the urine and the retention of desirable materials in the blood. So when you start out in life, the nephron, you have about 1 million of these units per kidney. So that's your nephron endowment, so to speak. So in the healthy human has about 1 million per kidney. The nephron is comprised of the glomerulus and also the tubular components, and you can think of them as sort of a filtration and processing component meant to handle or meant to take the early filtrate, the primary filtrate, all the way down to the waste product of urine by continual removal of water and other materials that the body would like to retain in the blood. And what happens when you have glomerular disease is that by some mechanism, whether it's sort of a toxic exposure, a genetic disease, some sort of insult of another type, obesity, virus, what have you. Ultimately, you end up with destruction or injury of the podocyte. That this cell that's in yellow here wrapped around those capillaries, like I said, they're very important in how the filtering capability of uh, the kidney gets its work done. And when the podocytes are damaged, all of that capacity or some of that capacity goes away. We see it clinically as protein that starts to come out in the urine because the podocytes are not there to present a barrier to prevent or retain protein. And what you might also see is some scarring. And that's part of what we mean when we describe FSGS as focal segmental glomerulosclerosis. So there's a spectrum of glomerular disease, and we're not going to discuss all of this, but what I really want you to take away from this graphic is that when we say glomerular disease, we're not talking about just one thing, that multiple routes can lead you to this final end result of podocyte injury. And in the context of AMKD, it's this entity here, FSGS, that seems to give us our most injurious consequence. So FSGS is a histologic finding. It's not really a disease, even though we kind of refer to it that way. Um, it is an injury pattern. So it's kind of what you see when you look through the microscope. You'll see a glomerulus, and then within that loop, that capillary tuft, you'll see areas of it that are scarred. You look across the kidney, maybe you see it in a sort of a patchy distribution, just depends, and you can see it in different areas. It really is sort of just the pathologist's description of what you're seeing. But the importance of it is that when you see it, you know what kind of problem you're working with. You can almost, in a way, kind of understand what type of clinical presentation may have come along with that histologic finding. So it's a very important diagnosis to be able to make. Unfortunately, we don't have a lot of very descriptive information beyond FSGS to provide. There are newer technologies that are allowing us a greater capacity for describing it. But the lesion, for all intents and purposes, is not as well understood as I think we would like for it to be. There's wide variation in the clinical presentation, obviously, depending on when in the disease course you are engaging with the patient, what is the cause, how rapid is the decline associated with it. All of that can be very different, all with the same label, FSGS. It's the most common primary glomerular lesion that causes kidney disease in the United States. And that's an important point to hold on to. That when you kind of look at the, excuse me, last 20 years, a lot of the glomerular disease that you can note in the United States is really going to be tied back to FSGS, however you got there. Okay. FSGS results, as I said, from podocyte injury. And at present, there are no selective therapies, but we're obviously working hard on that. So this is what a normal human glomerulus looks like, cut in cross section. So you're looking more or less down the barrel of those capillaries, and all of this sort of material that you see around it with the little black, almost sort of raisin-like nuclei. Those are podocytes kind of covering the outside. Some of that may be cells inside the capillary, but for those that are on the outside, those tend to be podocytes. This is what happens when you have some injury that drives the development of the histologic lesion. A lot of scarring. It doesn't involve all of the capillary structure, but in some territories, and that's why it has that character, or that it's characterized as focal in nature. So you get this segmental scarring. Those territories are no longer capable of doing the filtration and normal functions of the glomerulus as they would. You're going to see that again, as I mentioned, as loss of protein, but you're also going to see it, it's part of a syndrome, ultimately what you'll see. And so it's not just proteinuria, it's going to be associated with hyperlipidemia. You may see it with hypertension. You almost certainly will because of the kidney, its integral role in managing blood pressure, regulating blood pressure. When the kidney starts to fail, you'll also start to see issues with the management or the regulation of blood pressure. So your patients are going to come in hypertensive. They won't all have a nephrotic syndrome, which is a certain degree of proteinuria. Some will be considered subnephrotic, but generally speaking, they're all going to be resistant to steroids, and that's an important point. It's sort of vestigial, but when you deal with nephrotic syndrome in the pediatric population, it tends to be a variant on the spectrum of glomerular disease, something uh, known as minimal change disease, and that's responsive to steroids. So, in a way to differentiate it clinically, you'll describe things as steroid resistant versus not steroid resistant. FSGS tends to be steroid resistant. And so, that portends a more difficult prognostic issue because, after steroids, you're very often left with few treatments that are going to be effective in reversing this or slowing progression. It's really interesting because, again, in the same period of my training, this disease has really rocketed up, and we don't understand entirely why. A 13-fold increase is what I've seen in the last estimate. But, that's substantial. So it makes you think there's got to be something. It's genetic, environmental, something that's driving this that we haven't quite fully appreciated yet. But certainly we're all in our different ways on the hunt for this. And we do believe at the end of this acquisition of new knowledge, there will be the opportunity to treat. Um, we just sort of to give you a sense for what this looks like in terms of numbers and in patients. There's a study that was published in PLOS One. It's a scientific journal where they more or less looked at the veteran population. They did what we call a retrospective cohort analysis, and they wanted to say, okay, we're going to define FSGS by certain ICD-9 codes, which is sort of our disease coding system. And people who have had two or more of those ICD-9 codes in that interval of time that we're looking across, so that was back from 2000 up to 2020, we're going to say that those people have FSGS. And we're going to look at the incidence and prevalence. We're going to calculate those. And we're going to also look at other associated features so that we know what this population looks like and how bad this disease is. What I want you to just take away from this, especially the panels over to the left, that there have been, fluctuations in the incidence, and this is all comers, but there have been fluctuations in the incidence, but more or less steadily um, uh, over the past 20 years or so, there's been an increase in the prevalence. And that says, that there may be, a leveling off, which might reflect some greater understanding, some new therapies that we have available to us, but we're still not putting a dent in this in the ways that we need to. What was missing, though, and what stood out in several studies before this is sort of the final point that I sort of make here is that most of these patients in this group where the prevalence is increasing were noted to be white, non-Hispanic, or Black. So for African Americans in particular, that's a substantial part of the group. And in fact, one of the things I remember being taught as a fellow is that if you walk into your clinic office and you're going to see someone as with a new consult for proteinuria, um, if they're African American, about 80% of the time, you can be you would be right in guessing that this was an issue with FSGS, particularly if they were younger. And so a lot of people took note of that and thought, well, that sort of speaks to ancestry, and that might be some factor driven by ancestry that we're not necessarily detecting in the aggregate. Maybe we need to break these things out by race and get a real, a more descriptive sense of what we're seeing in the population. You can also see it reflected in other ways because you know what FSGS is like, you know what the patterns of the disease are, how it evolves over time. And usually, probably within the first 5 years or so of the diagnosis, you're going to see people to start to have really significant declines in renal function. Maybe out by 7 to 10 years, they're already end stage. So you can then look at uh, large populations of people that are end stage and try to see if those same differences in FSGS actually are reflected in the rates of end stage kidney disease. And lo and behold, that's what you see. Almost the same interval of observation. African Americans being this sort of teal blue line, they have a much higher incidence of end stage kidney disease than do other racial and ethnic groups. And the point to make here is that if this is the case, that there must be something ancestry at least contributing to this trend. And so then it became the job of those who deal in um, the realm of genetics and understanding sort of the molecular biology of a problem to really figure out with the tools that we have, can we identify a new genetic modifier for the disease that could account for this degree of difference? And as I mentioned, in 2010, Dr. Genovese and Martin Pollack at Harvard Med School made that initial observation that in a gene known to protect individuals from African sleeping sickness, a trypanosomal infection in sub-Saharan and West Africa, um, uh, certain variants in that gene actually could then leave you exposed or at risk, at higher risk for the development of what we call APOL1-mediated kidney disease, or the FSGS phenotype lesion driven by the contribution of those genetic variants. And it's a really complicated story. It's a really excellent read. There are a lot of reviews that have been really well done, particularly as information has evolved. I encourage you to sit down and read them. It tells a story for the ages. But in brief, you basically had a protein that was intended to protect your cells, your blood cells from trypanosomal infection. And the way that the body did that was to create a protein, APOL1, and that protein had a certain coding sequence as all proteins do. Well, that would have been about 10,000 or so years ago. Over the course of time, fast forward, say maybe 6,000 years forward, those same organisms, these trypanosomes, developed a workaround, so to speak. So no longer was APOL1 able to do its harm to those organisms and prevent them from infecting your body. They worked around it so that it was excluded from the mechanisms of shuttling that caused ultimately death and explosion of the trypanosome. And so they survived. Well, humans also can adapt. And if you fast forward a couple thousand more years, you will see the development of these two genetic variants that are at issue today. And they restored a measure of protection. So protection that had been lost was then restored, but by virtue of taking on these two genetic variants. Well, then you get to the period of the transatlantic slave trade. And that's where it's thought that a lot of this really took off and changed the character of the molecule entirely. What happened in that was that you move large numbers of people bearing these variants to a completely new ecological context. There is admixture and a number of different influences that result in a different way that the protein and its function behaves in this new environment. Unfortunately for African Americans, because all of these changes were happening in people of sub-Saharan and West African ancestry, this is a problem that uniquely affects us. And the fallout, the result of it is exclusive to us. And so when you consider patterns like what I showed you just before in the previous slide, the patterns of end-stage kidney disease, patterns of FSGS prevalence and incidence, that is why, or at least that's what we believe present or currently, that is part of why those differences exist. It is the contributions of these two genetic variants on the function of the molecule that ultimately lead to this predisposition to develop this kidney disease. And we still don't quite know why that is, but we have data to support that conclusion. So this is another sort of recent take on the epidemiology of the disease. And again, I encourage, this is a really fascinating read. And if you ever have enough time, I know it's hard to say to medical students, but if you ever have enough time to really sit down and maybe dedicate an afternoon, some of the best work has been done in the last 10 years, and it's out of a number of labs that focus almost entirely on this as a subject. So it won't be hard to find the papers. But if you could just read the evolution of understanding, you can see, really how far we can come very quickly. And it's an inspiring thing, an inspiring subject to gain knowledge in. This is, though, a graph from a paper published recently by an investigator, Opiemi Olabisi, a leading expert in APOL1-mediated kidney disease and sort of the biology of APOL1. He's actually a colleague of mine at Duke, and he sort of provided for JAMA Insight a perspective on APOL1-mediated kidney disease. What they provided here was sort of sort of a risk assessment for the high risk genotype. And for the high risk genotype, APOL1, by the way, behaves like an autosomal recessive pair of variants. So both genes, both alleles have to be affected to put you at risk, at the highest risk for developing this problem. So what they wanted to look at here was um, what is the effect of the high risk changes as it relates to each particular type of disease. So you can see non-diabetic, CKD, maybe it increases the odds ratio somewhat, but not tremendously. Very different than HIV-associated nephropathy or FSGS. Very different, much greater risk. And so that really gave us pause, and it made it stand out as something that we had to address right away. On the spectrum of genetic disease, APOL1 would fall among the common variants. So about 50% of African Americans have APOL1 variants. Not the high risk combination. That's a smaller proportion, but at least one allele is affected. So you're going to see a high frequency in the population, but a low effect size. And this is sort of another summary, again, from the Olabisi lab, more or less showing all the different mechanisms that they were able to determine in a really, a tour de force study, all the different things that they can demonstrate that this particular protein can do. It can cause loss of potassium, disruption of mitochondrial function, disruption of the ER, all sorts of things that are interesting to note, but also potentially therapeutically modifiable. And people are taking this type of information to clinical trials. This is just sort of a number of the clinical trials that are currently ongoing. Some are still recruiting, some have been completed. Among them is the Vertex trial of inaxaplin. Really interesting data, very compelling story that I think is really exciting, and I'll go through a little bit of that data.
Hi,
I
Dr.
also
Hall. Just wanted to give you a 1-minute warning.
Gotcha.
Yeah.
Also wanted to mention OP's study that is also ongoing, the Care and Justice study. This is the Vertex study, some of the great data. Based on what we come to understand about it, we know that it's you can modify the function of this particular molecule and get a reduction in proteinuria. Very exciting as an idea. I'm going to forego the next bit of discussion. But I just wanted to leave you with this. EMBARK is designed to get the word out about this. and to really educate the community and pair them nationwide with clinicians that are not only well versed, but capable of managing the issue uh, from the very beginning, thereby cutting down on the time to treatment. So if you just go by an understanding of this as time is kidney, you can see why this will be a very helpful intervention. So I think I've given you as much as you can get in one little bite about this. It's a really interesting story. To the medical students, I encourage you to um, keep motivated about nephrology and focus on this as a way to not only gain access to the population and how you can be helpful, but maybe to inspire your careers. Thank you very much.
Great. Thank you so much, Dr. Hall, for sharing. That was really helpful to have kind of a scientific background to what we're talking about today. So we really appreciate it. I'll give one question just because we're running out of time, but if anyone has any questions, put them in the chat and I can definitely get back to you later with any questions. But if students are interested in learning more about APOL1, what's a resource that you would recommend students to go to get more information?
There are plenty of places you can go. Certainly, you can visit our website on EMBARK. There are a number by now of really well done resources that are just available online, YouTube, all the social media platforms where people have given lectures, they've given brief expositions, they've created graphics and visuals. I was going to tell you about my own studies. But everyone is excited about this. It's a big issue in the nephrology community, so there are plenty of places you can go to get quite a bit of really relevant information real time.
Perfect. Thank you so much. And I'll touch base with you as well so we can get some resources to send out to everyone so they can, if you ever have some time, kind of take an afternoon like you recommended and do a deep dive into this really important issue. Great. Thank you so much, Dr. Hall.